Hey everyone. Yesterday we covered the fundamentals of RDT and some portion of enzymes. So today we will covering the restriction endonucleases. This is a bacterial enzyme that cut double-stranded DNA into a fragments by recognizing a specific sequence. That sequence is known as restriction sites. So the term restriction comes from the idea that bacteria restrict the entry of foreign DNA into the host cells. So we have example of restriction endonucleases, E. coli 1. So how it is written? This portion is taken from one genus letter and two from species letter. This one is taken from the strain name R by 13 strain. R from R by 13 strain. And the I is order of identification. So restriction sites or the restriction endonucleases cut only the double helical segments that contain a particular nucleotide sequence from 4 to 8 base pair nucleotide that is in length. So this is known as restriction sites or restriction recognition. So these are generally palindromic sequence. What are palindromic sequence? That when we read the sequence from 5 prime to 3 prime end is same that as of the complementary strand. So when the restriction endonucleases cut the nucleotide sequences, it is generally shown by the symbol, by this symbol. So restriction endonucleases results in the blunt ends or the sticky ends. So what are the blunt ends? It falls under the symmetric axis. And what about the sticky ends? These are the overhanging ends. It doesn't fall under the symmetric axis. We will be taking both the examples 5 prime G A T C 3 prime N 3 prime N C T A T A G 5 prime. So here the enzyme E. coli V comes and results in the blunt ends. And here we have 5 prime G A 3 prime N and 3 prime C T T A G 5 prime N. So here E. coli results in the overhanging fragments. It cuts here. So now we will dealing with some of the important terms. First one is star activity. So what is star activities? That there are some of the restriction enzymes which are capable of recognizing restriction sites which are similar, but they are not identical. They are not identical into their defined restriction sites. Another term comes here is isochiasmers. So these are the pair of restriction enzymes that recognize the same recognition site and also cut at the same location. So what is it? It has different enzymes, same restriction site, and also same cutting sites. So example come here is so see here they have same recognition sites and they also cut at the same cutting site. Another term come here is neoschismers. So what is it? It is a different enzymes same recognition site, but they cut at different cutting sites. So example come here is, see here, the restriction sites is the same or recognition site is the same, but they have different enzymes and they cut at different sites. You must remember all these examples. Next term is isochronomers. So they are different enzymes. They have different recognition sites, and also their cutting site is also different. So they produce some cohesive ends. Example comes here is SAU3A, BAMHI, so what happened? These are the different enzymes, different recognition sites, different cutting sites, but they produce same cohesive ends. So these are some of the important terms that we have gone through. Another comes one question that the restriction enzyme is a bacterial enzyme. So question come here is that why doesn't the bacteria degrade itself? So answer here is that bacteria protect their DNA by modifying their own recognition sequence. So how they modify? They add methyl group. So here are some of the position of methylation. First one is at the C5 carbon of cytosine, N4 amino group of cytosine, and N6 group amino group of adenine. So E. coli R13 strain which produce E. coli also make E. coli methylation. So it attach methyl group to the N6 position of the second adenine residue, where it add methyl group to the N6 position of the second adenine residue. So we have three types of restriction enzymes: type 1, type 2, type 3. So type 1 is bifunctional enzyme. Second is unifunctional, and type 3 is also bifunctional. So bifunctional here means they have both endonucleases and methylases activity, but unifunctional means they have separate endonucleases and methylases activity. So type 1 has 3 different subunits. They have 2 identical subunits, and it has 2 different subunits. So the type 1 is ATP dependent. Type 2 is ATP independent, and type 3 is again ATP dependent. So the type 1 cleaves random up to recognition sites from the 1000 base pair away from the recognition sites. Type 2 cuts at or near the restriction sites, and type 3 cuts at 24 to 26 base pair or to the 3' end of the restriction sites. So basically type 1 and type 3 restriction enzymes are not that useful because they cleave DNA sites other than the cleavage sites or the recognition sites. But type 2 cut at near or at the recognition sites. So it is basically used for cloning or mapping purposes because they recognize the specific sites and cleave just at these sites. So another we have and modification enzymes. So it makes changes to the end of the nucleic acid. So first one is TDT, terminal deoxynucleotide transferase. So what it is a template independent. It doesn't require any template DNA polymerase because it is able to synthesize new polynucleotide without any base pair of the incoming nucleotide. So it is basically used for the formation of cohesive end by homopolymer tailing. So what this does, it catalyzes the addition of series of nucleotide. What it does, it catalyzes the series of nucleotides onto the 3' OH of the double-stranded DNA molecule. So if this reaction is carried out in the presence of just one type of deoxyribonucleotide, a homopolymer tail is produced. We will sing one example. 5G. So in the presence of TDT, DCTP means cytosine nucleotide is added and a homopolymer tail is produced. So what happens is 3' ends are made. So next comes our alkaline phosphatase. So anywhere the term phosphatase comes, it means removal of phosphate group. So it is usually obtained from the sources E. coli or calf intestinal tissues. Second is alkaline phosphatase. Phosphatase comes, matlab, they results in the removal of phosphate group. So it is more stable, like the bacterial alkaline is more stable but is less active. I said that they are obtained from the E. coli and calf intestinal tissue, and bacterial one alkaline phosphate is more stable than calf intestine but it is less active. So another term comes is another enzyme T4 polynucleotide kinases. The term kinases means addition of phosphate group. Always remember, phosphatase means removal of phosphate and kinases mean addition of phosphate group. So it is the reverse of alkaline phosphates. So kinase obtained from E. coli results in infected with T4 phase performs a reverse reaction like of the alkaline phosphatase. It add phosphate to the 5' ends of the DNA sequences. So it is mainly used for the labeling DNA molecules. They use two type of reaction, that is forward reaction and exchange reaction. So what happens in forward reaction is that polynucleotide transfers gamma phosphate from ATP, the 5' end of the polynucleotide. ATP means there are three phosphate present, triphosphate, alpha, beta, and gamma. So this gamma phosphate is transferred to the 5' end of the polynucleotide. Whereas in the exchange reaction, they target the polynucleotide that has a 5' phosphate that is incubated with an excess of ADP. So So what happens in the exchange reaction? They target the polynucleotide that has a 5' phosphatase that is incubated with an excess of ADP, and it transferred the phosphate from polynucleotide to ADP and it makes it a ATP. So then it carries out a forward reaction and performs and or transfers a phosphate group from ATP onto the target polynucleotide. So that's all about today's lecture. In the next part, we will covering the vectors portion. Thanks for watching and please like, share and subscribe the channel.